Hi there, and welcome to the module of database retrieval using Biopython. In this module, we will explore how to use biological database that can aid in your research and analysis and demonstrate how Biopython can be used to retrieve your gene, mRNA, protein, and genomic sequence, your PDB structure from your PDB databases, and query and analyze your retrieved data. So first of all, NCBI is a vital biological database that enables us to retrieve gene information that helps in gene research analysis. We can easily retrieve the gene information using Biopython. This information is essential for wide range of biological analysis such as gene expression studies, mutation analysis, and much more. So here is the example code snippet for retrieving the information using the Biopython. So first of all, we are going to import our intras handle, and after that we are going to use this intras handle to retrieve the gene information. So if we look at the code that we are going to discuss here, we can see that we can just import the intras module from the NCBI database, and then we can create a function to fetch the genomic sequence using the provided accession number. So here we can give the accession. Here is the intras mail that you have to replace with your email address, and I have given my email address here. So we will send the email to allow NCBI to contact you if you have any issue with your query. And after that, are going to create some handles, which is efetch is used to fetch the sequence data from nucleotide database using the provided accession number. So retype the FASTA is the specific data format that should be returned in the sequence format, which is FASTA. And then we are going to remode the text, indicate the data should be returned as your text string. So we are going to read the fetched sequence data, and after that handle and close the resources, and then we are going to return the genomic sequence as a string. So here we can see example usage. We are going to give our genomic sequence, and then we are going to call the function with the accession number to retrieve the sequence, and then we are going to print the retrieved sequence. And so it depends on the internet how much time it is going to take. So it is taking a lot of time. So after that, we can see that we can also retrieve mRNA sequence. So mRNA sequence is a crucial intermediate in protein synthesis that carries the genetic information from the DNA to ribosomes. So Biopython allows us to easily retrieve mRNA sequence from NCBI with its powerful functionalities. mRNA sequence information is essential for understanding the gene expression mechanism, transcriptomics, and proteomics. So if we look at the code of retrieving the mRNA sequence, so first of all, we are going to import the intras module. Like that, we are going to call the function to retrieve by using the provided accession number, and we are going to replace it with our email, then going to use the same format that we have discussed above, and then just going to put our mRNA accession here, and then we are going to fetch our mRNA sequence by calling the function. And here we can see we got the mRNA sequence of our protein, which is Nexus topicales animal, and its mRNA sequence is given here. You can also copy this or just export it to a text file. So now if we move on to retrieving a protein sequence. So proteins are the functional molecules that perform a wide range of functions in living organisms. Biopython allows us to retrieve protein sequences from NCBI, which is necessary for studying protein structure and the structure and function, drug design, and other biological applications. So the code snippet that we are going to discuss for this is for retrieving the protein sequence. So just like that, we are going to import our intras module, and then we are going to call the function which is going to set a function to retrieve our protein of interest by using the accession number, and then we are going to call that function to retrieve the sequence, and after that we can print the sequence. So here you can see I have called the insulin a chain A chain sequence to see if it's retrieved that. So here you can see insulin human fold contains the rect name, and insulin B chain contains the insulin A chain sequence. So here you can see that. After that, if we move on to the genomic sequence. So genomic sequences complete the sets of DNA that constitute an organism genome. So with Biopython, you can retrieve the genomic sequence easily. These genomic sequences are essential for genome assembly, comparative genomics, and other critical areas of biological biology that require genetic information. So by using the Biopython, it is same. You have to import the Entrez module just like before, and then you are going to call some accession number, and after that changing that, the change that you want to do is call the function by using genomic sequence. So you are going to change it to genomic sequence, and then you can run this, and your genomic sequence will be called. So that was for the intras, that is for the NCBI. You can also import, download and retrieve your protein structure by using the PDB database. So the PDB is a repository of protein 3D structure which are essential for studying the protein structure and drug design. With Biopython, you can retrieve the protein structure from PDB quickly. So for that, what you have to do is you have to import the PDB list module from Biopython for accessing the protein database. And then you have to create a function to fetch the PDB structure using the PDB ID. And then you have to use the retrieve PDB method to fetch the PDB structure with the given ID. And then you have to define your PDB file format. And after that, you have to indicate that PDB file should be saved in your current directory. And the downloaded PDB file will be in the PDB format. And then you can just use an as example PDB1 CRAN. You can also replace it with the PDB ID. You have to retrieve the structure. So if we go to our database, and here we can see we have the one. So we can just look at what we have. PDB ID replace the PD1 CRAN. Okay, so here we can see if we have a structure which is named 1K. So if we are going to run this code again, so let's just find. Here you can see the file is downloaded. I'm going to just open this file in UCFC Chimera to see if it is a protein structure. So here you can see the file is now downloaded, and you can use this file. So here you can see that is a protein structure downloaded from Chimera. You can also search the database to validate this structure. So what else we can do? We can query the database with Biopython. So one of the significant advantages of biological databases is its ability to perform specific queries to fetch the relevant data. Biopython provides a powerful interface for querying the database by gene name, protein name, accession number, and other criteria. So this criteria enhance the data retrieval efficiency and accuracy. And here you can see that if we have how can we query the database? First of all, we have to import necessary module from Python, and then we have to enter our database email, and then we can use Entrez to retrieve the specific information, and then we can search for the nucleotide sequence given the name of CRT plasmodium, and if there are more results you can limit, you can also do that. Then we are going to retrieve the sequence IDs and then fetch the records of protein IDs that are in the GenBank and search for the specific that we have KM28867, and then we are going to analyze our sequence features, and then print the annotations of retrieved sequence. So we can also print the length of the sequence as well, because we are querying the database. And after that, we can retrieve and display the references associated with the sequence as well. So here you can see it is a result that are given here. You can check this result by analyzing it by yourself. You can also analyze the research data. For example, I have given a sequence. I am checking its GC content. So there is 30%, 34% GC sequence consisting in this amino in this sequence. We have already discussed it before. And then we can also filter the data. For example, by using the seq.io module from Biopython, we can also praise the sequence data. We can function and filter based on their length, and then create a list to store the filtered sequence. So example usage, we can see replace the file with your past sequence. I have given a past sequence, and then set the minimal length you want to. I have just set it to the 100 amino acid, and then you can just praise and call the function to filter the sequence, and then it will give you your filtered sequence. So that was is it for the database retrieval system. You don't have to worry about the code. All the code will be given on my GitHub repository, and that was it for this.